The Central Dogma, a project. Replication. DNA, or deoxyribonucleic acid, is made up of two interlocking strands consisting of nitrogenous bases connected by sugar phosphate backbones. These are related in a double helix structure. This DNA is held in the nucleus and contains the code for all of the proteins within the cell. In order to be turned into useful proteins, it must first replicate. The first step in replication occurs when the enzyme helicase comes through and separates the double helix into a leading strand and a lagging strand, each with a backbone and exposed nucleotides. These two are held apart throughout replication by a single strand binding protein. Further along the helix is a topoisomerase protein that helps to relieve stress in the helix from unwinding the strands. The enzyme primase then introduces a primer made up of several RNA bases to begin replication. DNA polymerase III adds bases off of this primer to correspond with the exposed original bases. The four types of nucleotides are adenine, thymine, cytosine, and guanine, A matching with T and C matching with G. The ends of the DNA strands are marked as either a 3' or 5' end depending on the number of carbons. The DNA is replicated from 5' to 3', but read 3' to 5', and the two strands run anti-parallel to each other so that one top end is 5' and the other is 3'. The leading strand is replicated in one piece while the lagging strand is broken into several pieces called Okazaki fragments. These fragments are created because of the anti-parallel structure, which requires synthesis from 5' to 3'. Without them, it would be impossible to replicate in this direction on the lagging strand. Each starting point, marked by a primer, is located at particular DNA sequences called origins of replication. Once all of the exposed nucleotides have been paired with a new corresponding base, the enzyme DNA polymerase I removes the RNA primers that started the replication process, replacing them with correct DNA nucleotides. This is also the point at which the strands are proofread for mistakes. If any are found, a nuclease cuts out the incorrect segment and fixes it. To finish the process, DNA ligase joins all unconnected pieces, such as recombination fragments and Okazaki fragments. Transcription. Transcription is the process by which DNA is rewritten as pre-messenger RNA, or pre-mRNA. The mRNA will eventually carry the code for a particular protein from the nucleus to the ribosome. The phase begins at the promoter, a specific sequence of bases that initiates transcription. In prokaryotes, an activator, the protein responsible for signaling the process to begin, binds to the promoter, stimulating the start of transcription. In eukaryotes, the activator protein binds instead to control elements, segments of non-coding DNA that act as binding sites for transcription regulation. These can be broken into two categories based on their location. Proximal control elements are found near the promoter, while distal control elements are farther away, and groups of the latter ones are called enhancers. In bacteria, a small molecule called the inducer allosterically inhibits bacterial repressor proteins, which are themselves transcription-inhibiting molecules, ensuring that the repressor cannot bind to the pattern of bases called the operator. The operator is responsible for controlling the synthesis of pre-mRNA by the RNA polymerase. The inducer's attachment to the repressor activates an operon, a unit containing related genes, the operator and the promoter, this activation allowing the process of transcription to go forth. Though in this case the repressor was inhibited, found in both prokaryotes and eukaryotes, repressors are sometimes allowed to function as they are the primary way to inhibit transcription. Transcription factors also affect the process. Repressors function as general transcription factors as they influence the entire matching mechanism, while other proteins act as specific factors that only impact a single gene. Mediators take the message from the transcription factors, which are bonded to control elements, and carry them to the RNA polymerase, making sure the orders are received. The main function of transcription lies in the pairing of RNA nucleotides off of one strand of DNA. RNA polymerase adds the correct RNA bases in the same way that DNA polymerase III adds DNA bases in replication, A with T and G with C, except that RNA does not have the base nucleotide thymine. Instead, every thymine is replaced with a uracil, so that A now pairs with U as strands of pre-mRNA are created. During transcription, the 5' end is capped to protect the RNA sequence from external damage, and once the process is complete, the 3' end is similarly finished with a poly A tail, a sequence of 50 to 250 adenine bases. This concludes the pre-mRNA primary transcript polypeptide. RNA processing. The next step turns pre-mRNA into mRNA during RNA processing. This begins with the capping of each end, occurring while the bases are still being transcribed. Next comes the removal of large sections of the strand due to the fact that they are non-coding sequences. These sections are called introns and are cut out by a large protein complex called a spliceosome. This complex is made up of many smaller parts, including small nuclear ribonucleoproteins, also known as snRNPs, and other proteins. The remaining necessary fragments of code, known as exons, are then joined together to once again form one continuous strand of mRNA. Sometimes proteins are not needed at all, as RNA molecules themselves can act as enzymes. These are called ribozymes, and prompt mRNA slicing and intron removal without help from protein enzymes. Though they aren't included in the final version of mRNA, introns do serve a purpose. Because DNA sequences can sometimes code for multiple polypeptides, what appear to be superfluous introns in one expression may in fact be functioning exons when coding for a different protein. Translation. Now that the mRNA has been created and modified, it travels from the nucleus to the ribosome, an organelle composed of ribosomal RNA, also known as rRNA, and protein. This is where proteins are manufactured through the translation of the mRNA sequence to an amino acid sequence. The mRNA, as we know, has millions of exposed base pairs made up of A, U, G, and C. These nucleotides are compiled into groups of 3 called codons, which code for one of 20 possible amino acids. In the ribosome, the mRNA is matched up with complementary tRNA bases. The tRNA is also organized in sets of 3 bases, known as anticodons, that are attached to an amino acid. The mRNA codons match with the tRNA codons, eventually creating a chain of amino acids which make up the protein that was originally being coded for. The ribosome contains 3 main sites that are involved in translation. The A site holds the tRNA segment that contains the amino acid that will be added to the chain next. The P site holds the tRNA attached to the growing chain polypeptides. And the third and final binding site is the E site, where tRNA that is no longer needed exits the ribosome. As the amino acids associated with the tRNA attach into their own long string, the protein is formed. Once completed, this exits the ribosome, along the way being folded into a specific shape based on the characteristics of its individual atoms. The hydrophobic parts fold in while the hydrophilic sections face out. This complicated process is assisted by chaperonin, a protein that helps other proteins fold correctly so that they can carry out their functions. If the protein built is destined for the endomembrane system or for secretion from the cell, then it contains a signal peptide, a sequence of around 20 amino acids near the leading edge of the complete polypeptide. This leads the protein into the rough endoplasmic reticulum. It's then cut away and the free protein is further processed and eventually moved to its final destination by a transport vesicle. The newly formed protein is then integrated to complete a specific task, finally completing the central dogma system.